12 enzymes. Almost all enzymes are protein. There are some nucleic acid that behave like enzymes. These are called ribozyme. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and the tertiary structure. When you look at a tertiary structure that is given in figure 9.4b, you will notice that the backbone of the protein chain fold upon itself. The chain crisscross itself, and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate feeds. Thus, enzymes, through their active site, catalyze reaction at a higher rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference need mention: inorganic catalysts work efficiently at high temperature and high pressure, while enzymes get damaged at high temperature, that is above 40 degrees Celsius. However, enzymes isolated from the organism who normally live under extremely high temperature, example hot vents and sulfur springs, are stable and retain their catalytic power even at high temperature up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. 9.12.1 Chemical reaction. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compound undergo two types of changes. A physical change simply refers to a change in a shape without breaking of bond. This is a physical process. Another physical process is a change in state of matter, when ice melt into water or when water become a vapor. These are also physical process. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of a physical or chemical process refer to the amount of product formed per unit time. It can be expressed as rate can also be called velocity if the direction is specified. Rates of physical and chemical process are influenced by temperature, among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed ones. When enzymes catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, in the absence of any enzyme, this reaction is very slow with about 200 molecules of H2 bicarbonate H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speed dramatically with about 6 lakh molecule being formed every second. The enzymes has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the step is catalyzed by the same enzyme complex or different enzyme, is called a metabolic pathway. For example, is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reaction. When you study respiration in chapter 14, you will study this reaction. At this stage, you should know that this very metabolic pathway with one or two additional reaction give rise to a variety of metabolic end product. In our skeletal muscle, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast during fermentation, the same pathway leads to the production of ethanol, alcohol. Hence, in different conditions, different products are possible. 9.12.2 How do enzymes bring about such high rate of chemical conversion? To understand this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called substrate. Hence enzymes, that is protein with three-dimensional with three-dimensional structure including including an active site convert a substrate into a product P. Symbolically, this can be depicted as S to P. It is now understood that the substrate S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of ES complex. E stand for enzyme. This complex formation is a transient phenomena during the state where the substrate is found to the enzyme active site. A new structure of the substrate called transition state structure is formed very soon after the expected bond breaking making is completed. The product is released from the active site. In other words, the structure of the substrate gets transformed into the structure of product. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural state between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this pictorially through a graph, it looks like something as in figure 9.6. The y-axis represents the potential energy content. The x-axis represents the progression of the structural transformation or state through the transition state. You would notice the two things: the energy level difference between S and P. If P is at a lower level than S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or a spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of this transition state is called activation energy. Enzymes eventually bring down this energy barrier, making the transition of S to P more easy. 9.12.3 Nature of enzyme action. Each enzyme E has a substrate S binding site in its molecule, so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociate into its product P, and that unchanged enzyme with an intermediate formation of the enzyme product complex. The formation of the ES complex is essential for catalysis. The catalytic cycle of an enzyme action can be described in the following step. First, the substrate bind to the active site of the enzyme, fitting into the active site. 2. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. 3. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bond of the substrate, and the new enzyme product complex is formed. 4th, the enzyme releases the product of the reaction, and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. 9.12.4 Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the condition which can alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, or binding of specific chemical that regulate its activity. Temperature and pH. Enzymes generally function in a Temperature and pH. Enzymes generally function in a narrow range of temperature and pH that is given in figure 9.7. Each enzyme shows its highest activity at a particular temperature and pH, called the optimum temperature and optimum pH. Activity decline both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroy enzymatic activity because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction arises. At first, the reaction ultimately reaches a maximum velocity V max, which is not exceeded by any further rise in concentration of the substrate. This is because the enzymatic molecule are favored than the substrate molecule, and after saturation of this molecule, there are no free enzymes molecules to bind with the additional substrate molecule. That is given in figure 9.7. The activity of an enzyme is also sensitive to the presence of specific chemical that bind to the enzyme. When the binding of chemical sets off enzyme activity, the process is called inhibition and the chemical is called an inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as the competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind. As a result, the enzyme action declines. Example: inhibition of succinic acid, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. 9.12.5 Classification and nomenclature of enzyme. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 30 subclasses and named accordingly by a 4-digit number. Oxidoreductase, dehydrogenase enzymes which catalyze oxidoreduction between two substrates S and S. Example: transferase enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S. Hydrolysis enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic C single bond C halide or PN bonds. Lyase enzymes that catalyze removal of group from substrate by mechanism other than hydrolysis, leaving double bond. Isomerase include all enzymes catalyzing interconversion of optical, geometric, or positional isomers. Ligases enzymes catalyzing the linking together of two compounds. Example: enzymes which catalyze joining of CS, CN, PO, etc. bond. 9.12.6 Cofactor enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactor are bound to the enzyme to make the enzyme catalytically active. In this instance, the protein portion of the enzyme is called the apoenzyme. 3 kinds of cofactor may be identified: prosthetic groups, coenzymes, and metallians. Prosthetic groups are organic compound and are distinguished from other cofactor in that they are tightly bound to the apoenzyme. For example, in peroxidase and in catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is a part of the active site of the enzyme. Coenzymes are also organic compound, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as a cofactor in a number of different enzyme catalyzed reactions. The essential chemical component of many coenzymes are vitamin. Example: coenzyme nicotinamide adenine dinucleotide and NADP contain the vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bond with side chain at the active site and At the same time, form one or more coordination bond with the substrate. Example: is a zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. Summary: although there is a bewildering diversity of living organisms, their chemical composition and metabolic reaction appear to be remarkably similar. The elemental composition of living tissue and non-living matter appear also to be similar when analyzed qualitatively. However, closer examination of reveals that the relative abundance of carbon, hydrogen, oxygen is higher in living system when compared to inanimate matter. The most abundant chemical and living organism is water. There are thousands of small molecular weight, that is less than 1000 Dalton biomolecule amino acids, monosaccharide, disaccharide, sugar, fatty acid, glycerol, nucleotide, nucleosides, and nitrogen bases are some of the organic compounds seen in living organisms. There are 20 types of amino acids and 5 types of nucleotide. Fats and oils are glycerides, in which fatty acids are esterified to glycerol. Phospholipid contain, in addition of phosphorylated nitrogenous compound, only 3 types of macromolecule, that is protein, nucleic acid, and polysaccharides are found in living system. Lipids, because of their association with membrane, separate in the macromolecular fraction. Biomacromolecules are polymer. They are made of building blocks which are different. Proteins are heteropolymers made of amino acid. Nucleic acid, RNA or DNA, are composed of nucleotide. Biomacromolecules have a hierarchy of structure: primary, secondary, tertiary, and quaternary. Nucleic acids serve as a genetic material. Polysaccharides are component of cell wall in plants, fungi, and also of the exoskeleton of arthropods. They also are storage forms of energy, that is starch and glycogen. Proteins serve a variety of cellular functions. Many of them are enzymes, some are antibodies, some are receptors, some are hormones, and some other are structural protein. Collagen is the most abundant protein in animal world, and ribulose, biophosphate, carboxylase, oxygenase, rubisco is the most abundant protein in the whole of the biosphere. Enzymes are protein which catalyze biochemical reaction in the cell. Ribozymes are nucleic acid with catalytic power. Proteinaceous enzymes protein Proteinaceous enzymes exhibit substrate specificity, require optimum temperature and pH for maximal activity. They are denatured at higher temperature. Enzymes lower activation energy of reaction and enhance greatly the rate of the reaction. Nucleic acid carry hereditary information and are passed on from parental generation to progeny. Thank you friends for listening this audio, and please post your comment if you want any kind of improvement. So मिलते हैं next video में, and stay tuned with biology critic and enjoy the knowledge. Bye.